So good afternoon, everyone. Thank you for joining this lunchtime seminar with an appropriate topic of microbiome. I'm Daniel Rosenberg. I'm at the University of Connecticut School of Medicine in Farmington, Connecticut, and I'm also the director of our colon cancer prevention program. And today I'm going to pose the significant question: can we manipulate the gut microbiome for cancer prevention? And this is really becoming a huge question in the field. Today, I'm going to cover 5 topics that are related to this fundamental question about the microbiome. What are the known associations of the microbiome with various cancer sites? I'm going to review the demographics and risk factors for colon cancer. I'm going to ask the question and pose the answer: how does the gut microbiome influence metabolism and biological actions of diet-derived food constituents? And I've selected several for that topic. I'll indicate how the microbiome affects molecular pathways leading to disease with several examples. And finally, discuss how the microbiome could be modified to reduce cancer risk. So this wonderful review from about 12 years ago from John Milner really posed the questions that I'm going to try to answer today to some level, which would be the microbes, the types and numbers, the community structure interacts with food components in both directions. And both of those food components and microbes interact with the intestinal mucosa, ultimately to affect cancer risk and tumor behavior. And hopefully by the end of today's lunch session, we'll have a better understanding about all these two-way arrows and interactions. So the microbial community structure in colon cancer is highly complex. Of course, the microbiome is the total community of bacteria that live in the gut and beyond. We use 16S RNA sequencing as a phylogenetic screen or survey to get an in-depth information about microbial communities that are present within, for example, the gut lumen. Metagenomics is giving us a little bit more detail, where we study microbial communities that are sampled directly from their natural environment, in this case, the gut or stool sample, without any prior culturing. So we get functional gene composition of microbial communities. And we know overall that the microbiota can increase or decrease cancer susceptibility and progression by very diverse mechanisms. On the left side, we have some mechanisms that are harmful, and on the right side in green are more beneficial properties of the microbiome. So on the left, we see that acute activating inflammation through pro-inflammatory cytokines, influencing genomic instability. They're actually examples of DNA damaging agents that are elicited from certain microbes. And finally, formation of damaging metabolites. And we'll go through a couple of examples shortly. On the right side, we know that some metabolites made by the microbiome, for example, butyrate, are actually epigenetic modifiers and can inhibit histone deacetylases. And I'll show you examples of that shortly. Finally, we know that we can generate beneficial metabolites, short-chain fatty acids, IPA, the bile acid UDCA, urolithins, short-chain fatty acids, quite a host of different beneficial metabolites. Even folic acid is being produced by the microbiome. So clearly there's a lot of metabolic action going on in these bugs. So the first question is the known associations of the microbiome and cancer sites. And this is a very rapidly evolving field. Today, we'll just quickly overview lung, prostate, stomach, and colon, but this is not to exclude many other organ sites that may be impacted by the microbiome. This very nice review article from Hubrex at the IARC in France was published last year in Cancer Epi Biomarkers. And what they did in this review article was to look at the number of articles linked with various cancers, and then the total number of subjects, cases, and controls that were found for the total number of studies run. I'm going to give you an example of airway microbiome and lung for impacting lung cancer, gastric microbiome, gut microbiome, and prostate microbiome in the next few slides. Again, all reviews. So the lung microbiota seems to have a fairly important and direct impact on lung carcinogenesis, probably through some effects on immunity and probably ultimately affecting immunotherapy, although this work is really rapidly developing. So if we look at a healthy lung environment, we see low bacterial biomass under normal circumstances, and gamma delta T cells are active and inhibiting activation of Th1, Th17 cells. T regulatory cells are blocking activation of macrophages, and we keep airway inflammation very, very low. However, in dysbiosis, or which we'll talk about defined in better terms later, during microbial dysbiosis, we see a change in the bacterial biomass where it increases within the lung. And we see decreased community diversity, then the presence of some rather pathogenic microbes. And ultimately, we find that Th1, Th17 cells are becoming very active. There's a downregulation of anti-inflammatory T regulatory cells, ultimately producing airway inflammation, which, if we have initiated foci of cells, we can actually envision malignant transformation. So this has nothing to do with gut microbiome. This is all directly within the airways. The microbiome and cancer risk was nicely reviewed by Wheeler and Liss several years ago. Here, the important take-home messages for prostate are that, of course, diet can affect the gut microbiome, which can have an impact on prostate cancer prevention and treatment, of course. But and that's through some very well-known mechanisms of LPS, inflammation, changing the immune system, fatty acid metabolism, even bile salt production could affect the prostate. However, one also needs to consider the genitourinary microbiome. And there we may have chronic infections as a result, viral genetic incorporation, anti-inflammatory viral signals, pro-carcinogenic bacterial products. So there's really a lot going on within the genitourinary microbiome that needs to be considered when we think about the pathogenesis of prostate cancer. Next, probably the most clearly defined association of altered microbiome is with the effects of Helicobacter pylori infection in gastric cancer. So we know that in normal circumstance, H. pylori negative situation, there's a predominance of Firmicutes and actinobacteria, Bacteroidetes, and a fairly low and controlled level of Fuso and Proteobacteria. However, in a H. pylori infection, one actually begins to see this enormous change within the microbiome, where Proteobacteria become prominent. And during this change, we start seeing alterations in the normic gastric mucosa through these different stages, ultimately leading to intestinal metaplasia, dysplasia, and adenocarcinoma. And what's being filled in along the way now are changes within the community structure as each of these stages progress. So very, very important and rapidly evolving field of gastric cancer. Finally, there was a very nice review article from Song in Gastroenterology 2020, looking at pathways by which environmental dietary factors could affect the intestinal microbiome and potential roles in colorectal carcinogenesis. I'm going to go into a lot of more detail during my talk today, but of course, one can consider lifestyle, obesity, physical activity, fiber consumption, red and processed meat, all important in affecting the production of different metabolites within the microbiota, which can then directly impact the intestinal mucosa. And depending on what's going on within the epithelial cells or within the stroma, many of these metabolites can actually be important drivers or inhibitors of colorectal cancer. And we'll get into more detail shortly. The next topic is to review very briefly the demographics and risk factors for colon cancer, which is the disease that I've been studying for the last several decades. This nice Globocam estimate of incidence and mortality worldwide for 36 cancers in 185 countries is shown in a very easy to understand pie chart. And what we can see is that for both sexes, incidence is highest for lung cancer, as is mortality. For breast, incidence is the second highest, and colorectum is third highest. Prostate comes next at 7.1%. But for mortality, lung is still number one, and colorectum becomes number two. This is the same in the United States as well as globally. Stomach and liver worldwide for mortality really ramp up. Of course, H. pylori, which is a major problem worldwide, as well as hepatocellular carcinoma, which is important environmentally in many parts of Southeast Asia, for example. So liver cancer really becomes important, although it's not unimportant in the United States. It's just not as highly represented. For colon cancer, how common is it other than skin? Colorectal is, again, the third most commonly diagnosed cancer in the United States. Estimates from the ACS for 2021 are 104,000 new cases of colon and 45,000 new cases of rectal. The rate of people being diagnosed with colon and rectum has dropped overall since the mid-'80s, and this is mainly from screening colonoscopies. And fortunately, people are really listening to their doctors and starting to change their lifestyles. From 2013 to 2017, incidence rates have been dropping about 1% a year. However, this protective effect is really reflected almost entirely in older adults, and it masks the rising incidence among younger adults that's been occurring in colorectal cancer since at least the mid-'90s. So from 2012 through 2016, early onset colorectal cancer has increased every year by approximately 2% in people younger than 50. The NCI is so concerned about this that they put out a provocative question about a year ago. We were fortunate to receive a NCI grant to study early onset colorectal cancer, and our work has already begun. Unfortunately, sadly, we're getting quite a few early onset colorectal cancer cases already in some of our local hospitals where we're getting samples from. Overall, the lifetime risk of developing colon cancer is about 4% in men and women. Risk factors are well known, well described. We know that there's several heritable genetic syndromes, FAP and HNPCC or Lynch syndrome, which are important for cancer risk, but probably account for less than 5%. Personal family history is a fairly important risk factor for a first degree relative less than 60. Then there are quite a number of other risk factors: tobacco, alcohol, longstanding inflammatory bowel disease, lots of things that contribute to a metabolic syndrome, increased BMI, fast food syndrome, and finally, dysbiosis, which of course we're talking about today. On the positive side, decreased risk occurs from prior endoscopies. So keep getting your colonoscopies every 5 to 10 years, depending on what they find. Calcium vitamin D is a question mark. My laboratory has been studying vitamin D and its actions on the colon for quite a few years. And there are still a lot of questions as to why vitamin D is not 100% proven as an effective chemotherapeutic. We think we have some answers to that related to the vitamin D receptor, and we're working very hard on that. Omega-3 fatty acids such as EPA and DHA are clearly beneficial. NSAID use is now thrown into disarray again with the recent warnings that aspirin use in people over 70 years of age could be problematic in terms of cardiovascular events. And so now we don't know what to do with recommendations for aspirin. Estrogen is fascinating. Women do have an increased risk of certain subtypes of colorectal cancer. It could be related to perimenopausal changes. And however, estrogen would not ever be used as a chemotherapeutic agent because of its obvious effects on cancer. There's incomplete data on folate, vitamin E, natural food components, such as the Ellagitannins, and I hope I can bring up more of this detail later in the talk. And you can see some of the things that are beneficial or under intense study. And on the right side, you'll hear more about walnut, which is also appropriate for a lunchtime talk. Third topic would be, how does the gut microbiome influence the metabolism and the biological actions of diet-derived food constituents? And amino acids, fiber, ellagitannins are provide excellent examples of microbial human co-metabolic pathways. And let me explain what I mean by that for tryptophan. So tryptophan is broken down by the microbiome into quite a few different metabolites, most of which have beneficial properties on the intestinal mucosa. For example, indole IPA have very, very positive effects on tight junction regulation. And some of these metabolites have antioxidative properties. So tryptophan is clearly being utilized by the microbiome to generate largely beneficial metabolites. The opposite occurs with choline. Choline found in eggs and meat and dairy is metabolized by, and here in 2017 in this fascinating Copal Review article in Science, we still didn't know which microbes were assigned specifically to the choline metabolic pathway. However, choline does produce several disease-associated metabolites, TMA and trimethylamine and oxide. TMA is generated by microbiome, and then after it's systemically absorbed, it goes to the liver where it can be converted to TMAO. And TMAO is associated with cardiovascular diseases, NAFLD, chronic kidney disease, and IBD. So again, this is an example of metabolism into non-beneficial substances. Now, short-chain fatty acids and butyrate have been known to be a positive beneficial pathway of microbial fermentation for many years. And in fact, Charlie Jardine at UConn Storrs published this paper in Gastroenterology over 2 decades ago, where he actually began to figure out how butyrate was actually causing a beneficial effect. He argued that butyrate was inhibiting HDACs, and as a consequence of HDAC inhibition, there was a reduction in NF-kappa B activity, which is a very pro-inflammatory mediator. And that reduced a number of different pro-inflammatory cytokines, which would attenuate colitis and other inflammatory diseases. Now, short-chain fatty acids, I just want to say, are much more complicated than simply butyrate. They include acetate, propionate, and actually a number of other minor short-chain fatty acids that have properties that are just now being defined. More about the butyrate and dietary fiber protecting against colorectal cancer in a microbiota-dependent manner. We know that under normal circumstances, butyrate is a primary energy source in normal colonocytes, and we can see butyrate being generated from fiber through the microbiome, and butyrate then entering into the TCA cycle, and everything is happy in the colon, generating lots of nice energy. However, in a colorectal cancer cell where everything changes metabolically, glucose becomes a primary energy source. However, if you can load up with enough butyrate, the butyrate will be inhibiting the HDACs, which can block some of the pro-apoptotic and cell cycle regulatory genes, have very, very beneficial effects on colorectal cancer progression. This is great, and everything is wonderful, and eat your fiber, until a paper just came out fairly recently from Japan, from Matsushita et al. In Cancer Research, which is now proposing that short-chain fatty acids from the microbiome may actually promote prostate cancer growth via IGF-1 signaling. So this is really a very, very confounding observation. And now the field really needs to sort out how to handle this type of new information about prostate cancer promotion. Finally, my last subtopic within dietary constituents and metabolism would be ellagic acid, which seems to all be good so far. I haven't really found any adverse effects of the ellagic acid and its metabolites. So ellagic acid is in many foods. We're going to go into great detail shortly. The stomach releases ellagic acid from the precursors that are found in fruits and nuts, which in vegetables, which are the Ellagitannins. And then ellagic acid is converted by the microbiome into a number of different urolithins. Urolithins and have all sorts of effects on reducing inflammation and having very beneficial effects on different organ systems in the body. And I'll talk quite a lot about this and give you some preliminary data from some of our studies shortly. My fourth topic is to indicate how the microbiome might affect molecular pathways that lead to disease. And I'm going to give you 3 examples for which there's probably the most evidence: Fusobacterium, Enterotoxigenic E. coli, and Bacteroides fragilis. So colorectal cancer is often associated with dramatic alterations in the microbial composition of the tumor and adjacent mucosa. And we call this dysbiosis. And in some cases, there are increased concentrations of pathogenic bacteria. But there can also be situations where there's an absence of certain protective or beneficial strains. And this was very nicely reviewed by Tilg and Cancer Cell in 2018, and a beautiful review article from Cynthia Sears that just came out quite recently, Cancer Discovery. So I recommend you reading both of these review articles to really learn about this topic. Dr. Sears originally proposed the idea of the alpha bug, which would be postulated not only to induce tumors directly, but also to remodel the bacterial community to one that promotes the ability of an alpha bug to induce intestinal mucosal responses. And these could be direct epithelial changes or immunological changes, ultimately leading to cancer. And Dr. Sears has suggested that the alpha bug may crowd out cancer protective bacteria, such as Clostridium or Thermophilus. I've put asterisks next to the 3 bugs that I'll talk in a little bit more mechanistic detail about in the next few slides. And the other idea that has been proposed is the driver-passenger model, which is a counterpart to the well-known genetic driver-passenger model for colorectal cancer, where we classify microbes into 2 categories. Certain indigenous bacteria, which are drivers, could produce genotoxic substances that directly damage host DNA, initiating colon cancer. And certain environmental alterations might favor proliferation of opportunistic bacteria, which are passengers that could mediate tumor formation. In this case, Fusobacterium, which is a complex group of bacteria that actually are common and colonize the oral mucosa. But in some cases, the Fusobacterium nucleatum escapes and ends up finding an environment in the gut that is favorable for its growth. The tumorigenic mechanisms of Fusobacterium are complex and have been widely studied. There's both an increase in tumor cell proliferation. This may be occurring through activation of Wnt signaling, upregulation of some of the downstream targets of Wnt, such as c-Myc and cyclin D1, suppression of anti-tumor activity of natural killer cells and T cells, so an effect on anti-tumor immunity, generation of pro-inflammatory microenvironment, a number of cytokines that seem to be upregulated, associated with Fuso. Finally, promotion of chemoresistance, which is certainly a significant side effect of Fuso infection. The Enterotoxigenic Bacteroides fragilis and PKS E. coli are 2 other organisms that have been studied most intensively and do have some mechanistic information. So the ETBF seems to coat the tumor and recruit other bacteria to create what is referred to as a biofilm, which is a kind of a secreted layer of matrix that certain bacteria seem to favor and grow in. And what happens when this biofilm coats neoplasia, they attract immune cell recruitment and pro-inflammatory microenvironment, which may drive tumorigenesis. On the other hand, the PKS E. coli produces colobactins, which upon activation can actually form quite stable DNA adducts, which have been found in colorectal cancers. So although there's not any clear proof yet, and there are limitations in some of the animal modeling for these microbes, these 3 do seem to have the best evidence. And this was reviewed by Wendy Garrett in Science in 2019, which is a really good read. My last topic will be to discuss how the microbiome can be modified to reduce cancer risk. And again, this is really the holy grail now of what we're trying to do in terms of cancer prevention. So I will pose the question, can we modify the microbiome to produce a healthy cancer preventing community of microbes? And that would, of course, necessitate eradicating pathogenic bugs, such as F. nucleatum, and producing growth of healthy bugs using either pro or prebiotics. We know that in this very nice figure from Mills and Nutrients 2019, we're comparing poor quality diet and healthy diet and the consequences on the microbiome. So if we look at a healthy diet on the right, fruits, vegetables, fiber, etc., omega-3 fatty acids, we have high microbial diversity, it's colonization resistance, there's a very nice immune homeostasis going on, nothing is out of balance, healthy mucus layer, the gut barrier is healthy and functional, and there's high levels of short chain fatty acids. In a poor quality diet, let's say a Western diet, where we have a lot of animal-derived proteins, fats, refined grains and sugar, alcohol, corn-derived fructose, we're seeing reduced microbial diversity, pathogen infection, inflammation, erosion of the mucus layer, and intestinal permeability. Clearly a problem. In fact, today, the NCI reported on a study from Cornell looking at fructose and how fructose may actually be affecting colon cancer stem cells, cancer stem cells within the colon. And this is a very interesting study published in Nature just within the last few days. So clearly there's a lot of evidence for poor quality diet and what it might be doing both to the microbiome as well as to the intestinal mucosa. What strategies can we use to create a cancer preventive microbiome? This review from Oncogene in 2020, Winnie Fong has postulated some very interesting approaches, and I really like this review. It was a really nice article. The approaches being probiotics, prebiotics, postbiotics, which are metabolites generated from the microbiota, antibiotics, FMT, which is fecal microbiome transplant, or targeted microbial transplantation, all approaches that could be used to correct the microbiota composition, modulate immune system function, enhance barrier function, and prevent pathogen colonization. The outcome being a healthy cancer preventive microbiome, which is what we're all shooting for. So thinking about how to do this and how to study this, it's very challenging in human populations to really get at mechanism. And so, although I will show you some of what we're doing towards the end of the lunchtime seminar, first I'm going to provide some evidence that has been generated in mouse models that provide a powerful experimental system to study environmental dietary factors that influence the gut microbiota. So many factors affect the human gut microbiome, and this is on the left. And obviously this adds to an enormous amount of noise, which makes it very difficult to interpret results. However, mice are attractive for studying host genetic microbiome interactions because we can really control their environmental variation. Now, this has pluses and minuses, and one could argue that that's really not reflective of the real world, and I would agree with you. And you'll see the information we generate both from our mouse modeling as well as from our clinical studies. This is a study that was done about 5 years ago. Masako Nakanishi, who is a outstanding scientist, and this work was published in Cancer Prevention Research in 2016, using a strain A mouse treated with a chemical carcinogen to induce cancer. She followed that with another mouse model using an APC delta 14 mouse, which is a model of familial adenomatous polyposis. And what Masako found was that after 16 weeks on walnut, there was a significant reduction in the number of colon tumors in male mice, not so much in female, both in number and in size and volume of the tumors. It was really a remarkable effect, and it seemed to be most efficient at 7% walnut within the diet. That seemed to be the sweet spot. Masako looked at a number of different tumor-related biomarkers and couldn't really find anything that might explain why the walnut was having this preventive effect on colon cancer. And it was really puzzling, and we just couldn't figure out what's going on. At about this time, the state of Connecticut decided to helped the Jackson Laboratories, which is shown by an arrow here, build a new facility on our campus. And this is the main campus here. Here's actually my lab over here. And here's the Jack's Genomic Medicine, which is sort of a satellite campus from their Bar Harbor mouse group. And this group focuses more on human genomics. So at the time, one of the very first recruits there at Jack's back in 2015-16 was George Weinstock, who was a renowned microbiologist. And I hooked up with George to start to look at more detail at what's going on in these colons that we can't explain by classical biology. And George urged me to start looking at the microbiome. And so we did. And in that study that Masako ran, first we did a cluster analysis of the top 100 OTUs, operational taxonomic units, and it's shown as a heat map on the left. And what we found was that the OTUs, or the groups of microbes that group together into fairly tight knit groupings, depending on environmental changes, were actually grouping into 9 major phylogenetic clades. And clades are, again, another subgrouping of bacterial species. So we found 9 different clades, A through I, in this study of walnut, given diet through the diet on, I forgot to mention it, the walnut was included, added to a diet, a total Western diet. So it was a diet that had a number of properties associated with the Western diet. And the first thing we found doing the bioinformatics analysis was that OTU cluster E, which is over here, was strongly associated with carcinogen treatment, again, azoxymethane. And what Masako found was that clustering fell markedly as we increased the amount of walnuts. So something was going on that was being reduced in that group of microbes by walnut. Following the different clades, we found that there was actually a reduction in clade F might be an important event contributing to suppression of tumor initiation when going by numbers. So we see a positive correlation in clade F with the number of tumors. And then clade I, we found a negative correlation between abundance of this clade in tumors that were of larger size, 2 millimeters in diameter or larger. So we postulated that clade I, or that group of microbes affected by walnut, might actually be necessary for tumor suppression. The work was followed recently, about a year ago, through another study where we began to look at enterotypes. And this work was published in Cancer Prevention Research in 2020. Yanfei Chen was the first author of this study. And you can see George Weinstock, again, working closely with my laboratory to identify enterotype-like clustering going on through carcinogen, Western diet, and walnut. Those are our 3 environmental factors that we're trying to sort out. And what we found in these PCA plots were 3 specific enterotype-like clusters, 1, 2, and 3. And what we found is that walnuts seem to reduce risk of colorectal cancer in this strain A mouse eating a Western diet by altering microbiome. And the enterotype 2 does seem to have some protective effect on overall tumor numbers. So this work, again, was published about a year ago. So this is all well and good for the mouse, but really we're more interested in the human ultimately. And so I will shift now to some work that we've been doing with the human microbiome. The first few slides I will show you are from a recent paper that was published again with the Weinstock Laboratory, and Bo-Young Hong, who was a postdoc at the time, who's now an assistant professor in El Paso, Texas, at UT El Paso. What this study was intended to do was to characterize the adherent microbiome, which would be the microbiome not within the fecal stream, but actually fairly tightly adherent to the mucosal surface. And so the study population we looked at were all screening colonoscopy subjects, all healthy controls. They were either subjects that were free of ACF, which are aberrant crypt foci, or a very early precancerous lesion in the colon, or subjects, and polyp free as well, or subjects that had at least an ACF in the right colon, their proximal colon, but were polyp-free. And then subjects that had an ACF, one or more in the right colon, with some number of polyps present. So these were our three groups in this study. And what we found was that there clearly was an increase in the presence of adherent populations of microbes shown by this EUB 333 Psi3 staining, which shows an increase in punctate staining of unknown bacterial species in the ACF. And we found the same thing when we looked at polyp tissue as well. Now, what Bo Young found was that there was a significantly abundant bacterial taxa were stratified by the host somatic mutation and also microbiome profiles of all the samples. And to just show you what happened with unsupervised hierarchical clustering, we found two distinct clusters of microbiota. And those are shown by the cyan on the left and the magenta on the right. And so clearly the signatures of the microbiome clustered into these two groupings. Remarkably, and we're still working very intensively to try to understand what this is all about, we found that the presence of a somatic mutation within the epithelial cells themselves, measured by a process called DNA mass array or sequenome, which gives you very sensitive sequencing of somatic mutations, we found that those ACF that had an APC mutation, one of three different APC mutations, tended to have an adherent Intestinobacter species associated at a very high significance within the mucosa, whereas those ACF that had a KRAS mutation tended to have Ruminococcus navis strongly associated with the adherently within the mucosa. What this means, we do not know yet. Which way does this work? We don't know. In other words, is the specific colonization driving the somatic mutation, or is the somatic mutation attracting a particular colonization? We don't have an answer yet, but we're really interested in that observation. The other thing that was very interesting in this very complicated looking slide is when we took these two clusters, microbiome A and microbiome cluster B, which I showed you in the previous slide, and when we began to stratify the microbiome clusters according to the different groups, the no polyp, or we actually had the polyps broken down into hyperplastic polyps or even more advanced tubular adenomas and sessile serrated adenomas, we found that there was actually a distinct clustering of microbiome cluster B in those more advanced lesions. And in fact, we actually found a separation of a specific group of bacteria that seemed to be strongly associated with the presence of sessile serrated adenomas. This is very, very interesting to us. And in fact, I'll talk about it towards in the next 10 minutes before I finish my talk and what that may mean. So now that we've built up some basic knowledge of early neoplasia in screening patients in our population, we decided to run a clinical study, which was run by both Dr. Burke and Dr. Devers, both gastroenterologists that have been working with me for many years. John is actually the division chief of GI. Dr. Devers, who was a very deep friend and colleague, close friend and colleague, passed away last year. But these are the various groups and people that have been working together to actually run these clinical trials that I'm going to show you in the next few slides. So we've taken a whole food approach to modify the pathogenesis of colon cancer risk, and we focused on walnut. Walnuts have an amazing array of bioactive constituents. They're rich in omega-3 fatty acids, ALA, of course, rich in phytochemicals, including ellagitannins, a polyphenol, and also a number of different antioxidants. And so walnut constituents, we believe, are affecting the microbiome, and probably the arrow goes in two directions, which generate metabolites, which affect host biology, which we believe may be reducing cancer risk. Ellagic acid is the constituent that I am most focused on at the moment. Ellagic acid is a natural antioxidant phenol. It's found in many fruits and vegetables, pomegranate, raspberry, strawberry, and then some of the tree nuts. and it has anti-proliferative and antioxidant properties. This nice review from Ismail and Toxins shows you some of the cancer sites that Ellagitannins have been associated with in terms of protection or prevention. So Gary Stoner, one of the actual fathers of the field of chemoprevention, who published amazing papers on natural products, foods. Of course, everyone knows he was really interested in the black raspberry, but he was also interested in all sorts of polyphenols. And I put this paper up because in 1989, Gary was really thinking a lot about ellagic acid. And this paper is worth reading. Part of it is the historical perspective that Gary puts into the ellagic acid, that even back in 1960s, ellagic acid was being tested in animal cancer models. So the field has been around for 50 years. People have been thinking about ellagic acid and its metabolism for a long time. Ellagic acid is so common in so many foods, as I said. This Lipinska review article actually summarizes the concentrations of ellagic acid in different food products, and also the form that it comes in, which is typically in some type of an ellagittannin precursor. For walnut, the ellagittannin is pedunculagin. For raspberry, it's several other ellagitannins, pomegranate, punicillagin. So the complexity begins really in the whole food itself in terms of the actual form that the ellagic acid is present in. But one thing is certain, and this nice review from Landete in Spain gave a nice explanation of how the pedunculagin, for example, as one of the ellagitannins, is converted by intestinal pH to ellagic acid. So we go from the complex form to the ellagic acid free form. And then, of course, the microbiota begin to metabolize the ellagic acid into the urolithins, which we'll get into some further detail. One interesting thing from this Landeta review was that as ellagic acid is metabolized by microbiota into the different urolithins, culminating in urolithin B, which seems to be at the end of the metabolic pathway, there's an increase in lipophilicity and systemic absorption, which is good. However, there seems to be a decrease in antioxidant activity, which is not good. So we're trying to take these observations into mind when we're studying the various metabolites of urolithin that we're screening in our clinical trial. So I'm going to present some data from our pilot clinical trial, which was recently completed, and the analysis is nearly done. The more recently funded NCI trial through the FOA on pre and probiotics actually launches today with our first 2 patients. So, but the data I'll show you is from the pilot study. And it's a simple schemata where patient comes in as consents to a 7-day washout period where we try to get the patient off of any ellagic acid containing foods. At the second visit, we begin the walnut intervention. It's 3 weeks at 56 grams per day of whole-shelled walnut provided by the California Walnut Commission. At the last day of the intervention, at the first day, we collect blood, urine, and fecal samples, and then the patient gets to have a colonoscopy, and we take biopsy specimens up to 8. So this is the design of the studies. The hardest part probably for the patient is the avoidance of ellagic acid containing foods. Again, cranberries, strawberries, walnuts, grapes, almonds. It's hard. The patient has to avoid these foods for 4 weeks, but they're paid to be in the study, and they get to eat delicious walnuts. This pilot study was run under a clinical trial. And again, it was funded by the American Institute of Cancer Research, co-funded with the California Walnut Commission. And again, I thank both agencies for their incredible support and generosity in these studies. The metabolism of dietary ellagic acid is much more complex than you saw in the Landete review. In fact, our collaborator and colleague on our NCI grant, Juan Carlos Espin in Murcia, Spain, has been studying ellagic acid metabolism for quite a few years. And in a number of review articles, including this recent food and function study, Juan Carlos presents a very complex scheme for urolithin metabolism. Again, ultimately ending in urolithin B, although many of these arrows may go in several directions. And what we're trying to figure out, as is Juan Carlos, is which microbes are responsible for each one of these steps. And as you can imagine, that's an enormously complicated question to ask. So Gordonobacter, two different species of Gordonobacter seem to play a role in some stages, Ellagibacter in other stages, Bifidobacter in other stages. The only way to truly prove these different metabolic capacities is by isolating, culturing, and testing the metabolism of ellagic acid and the urolithins in the cultured conditions, either individually or with co-cultures of a number of bacteria. Our first results from 16S RNA sequencing show you in a heat map at the genus and OTU levels before and after walnut, kind of almost a disappointing finding in our first 40 patients, where we found that the walnut effect was not easily reflected by this heat map, which told us that the gut microbiome is actually personal, and the patients, individual patients before and after walnut are shown by color going down the right side. Walnut consumption did not seem to have a marked effect on the overall bacterial community in the human. It wasn't as obvious as what we saw in the mouse. However, there's always a however. We are beginning to tease apart that heat map. And this is all unpublished data. Dongbin Tran, who is a postdoc, a bioinformatics computational biologist in the Weinstock lab, has begun to bang away at these 16S RNA sequences data, as well as generating metagenomic data as well. And one of the first things she's finding is that Prevotella, which is actually a short-chain fatty acid producing microbe, which is associated with fiber and Mediterranean diet, is increased after walnut. And I've highlighted the green Prevotella over here. And that's kind of an interesting finding. It's not simple at all, even just reporting Prevotella, because there's different strains of Prevotella, different species of Prevotella. One strain of or species of Prevotella was shown fairly recently to actually have inflammatory effects in the colon. But as Dongbin cautioned me, that was not the same species of Prevotella that we've identified. So even when we find genus in species, we still don't really know exactly what we have in hand. Further, going along on our study, on our pilot study, we've been measuring 9 different metabolites of urolithins in the urine before and after walnut consumption. And what we're finding is that our first hypothesis, which is that feeding people walnuts will produce urolithins, is happening. Here I'm showing you the levels of urolithin A and isourolithin A, and we're seeing that before and after walnut. In almost every case, there's an increase. Also, when we add up all of the 9 metabolites, we've seen the same sort of trends. What is remarkable, and none of this data has been published yet because we're still analyzing it, is that there are patients that make 0 urolithins, even after 3 weeks of walnut and lots of ellagic acid in their diet. They don't have the right microbiota to make urolithins. And that's where the whole idea of the pre and probiotic comes in. So we're very interested in those patients that make none. And we also have some patients that make super amounts of urolithins. Those are the super producers. And of course, we're trying to isolate microbes from these patients to figure out what actually is accounting for that. Among the bacterial changes that we're seeing in this pilot study after walnut, OTU23 seems to be a good candidate. It increases after walnut, as you can see before and after, and it seems to be positively and significantly associated with levels of several different urolithins. And so again, here we see before and after. So this is a Lactospiraceae unclassified, so we don't really know too much about this particular genus, but we do know that it does seem to track, this OTU does seem to track with the ability to make urolithins. That's all our pilot data. In our newly launched NCI study, we are going to recruit 200 patients. Probably half or maybe more than half will come from the UConn Health Center. The other half will be coming from a group run by Steve Lipkin at Weill Cornell in New York City. So we're getting the patients from 2 different sites. And what we predict, we're using 2 criteria to recruit our patients in order to increase the probability of getting advanced adenomas. So we're using both elevated risk based on family history, personal or family history, which will increase quite a lot the likelihood of an advanced adenoma, as well as a positive fit test. And so what we're hoping to find will be lots of people with a nice, shiny, healthy colon. This was actually a walnut consuming patient. And then some number of patients with traditional polyps, and then some number of patients that have serrated polyps or SSAPs. And those are, we'll take biopsies from these patients after walnut, of course. And then what we will do with those biopsies, this is our workflow for what we typically do for aberrant crypt foci or sessile serrated adenomas, collect biopsies. We do laser capture microdissection. In some cases, we isolate organoid cultures, which we will be doing from the walnut exposed patients. And then we have a number of different omics analyses that we apply to these biopsy specimens in order to fully define what's going on in those lesions. Finally, our study will end with fecal microbial transplants in two different mouse models that we have in our laboratory, one of FAP, the other of SSA. The FAP, of course, using an APC mutation, the BRAF V600 mimicking the SSA. The APC mice we got from Dr. Eric Fearon, who's the director of the Comprehensive Cancer Center at the University of Michigan. And he generously provided with a conditional APC model, which we've been using for the last 2 years in several different studies. The idea is that we will isolate fecal or microbial samples from patients on the trial that are having a high likelihood of making urolithins, transplant them into the mice, feed the mice ellagic acid or some of the urolithin metabolites by either diet or gavage, and then look for effects on tumorigenesis. So that's our plan. And finally, my last slide to summarize and conclude everything. Walnut loaded with Ellagitannins, as well as a number of other healthful constituents, gamma tocopherol, omega-3 fatty acids, fiber, melatonin, phytochemicals, as I've been talking about. The ellagic acid is released in the stomach from Ellagitannins. Microbiome converts the ellagic acid into urolithins. The urolithins will then have effects on the host, and we're measuring again in the urine routinely. However, the microbiome may be interacting in a way that deeply affects the ability of the specific bugs to make urolithins. And some of the different walnut constituents may actually be affecting this ability of microbiome by changing the community structure of the microbiome, ultimately affecting the host. And some of the readouts we will use in addition to the colon biopsy specimens will be measuring short chain fatty acids, bile acids, and as well as cytokines in the tissue, in the blood, as well as in the fecal stream. So that is what we're hoping to ultimately find in this study. These are not actors. These are real human beings. This is Young-Jun Suhr, who's a very, very well-known chemoprevention expert in Seoul, South Korea, and his longtime colleague, Dr. Na. We were out. This is the last time I went out in real time, pre-COVID, to actually give a talk to human beings and not a computer screen. And Young-Jun was shocked at the amount of red meat that I was eating, but they loaded us up with all sorts of healthful Ellagittannin containing foods. So I think the damaging effects of the red meat were overcome. And I will just acknowledge the terrific support from the NIH, the California Walnut Commission, the American Institute of Cancer Research, Thermo Fisher and Thetis Pharmaceuticals, and finally to thank Dr. James Devers, my long-term colleague and collaborator who passed away unfortunately, but was an excellent colleague. And thank you very much for your attention.